All right, good morning. So I'm going to go ahead and record this lesson. So just give me like a couple minutes to run through it, and then I'll go ahead and stop recording. If you got any questions, let me know. All right, so I'm going to go ahead and go over cell division, so basic cell division, the cell cycle, mitosis. So before I get into all of that, so most of what you're going to learn about in this lesson is on eukaryotic cells. So I just want to touch base really quick on prokaryotic cells. So prokaryotic cells obviously divide too. They just do so through a process called binary fission. There's really, you don't need to know much of the steps to it. All you really need to know as far as like your end of course exam and stuff is that the bacteria basically they kind of elongate, um, the DNA replicates, it splits in half, and that's pretty much it. You know, as long as you know the name binary fission is for prokaryotic cells or bacteria, you should be good. All right, so now everything else is going to relate to, for the most part, it's going to relate to eukaryotic cells. So as far as the cell cycle goes, there's a couple main parts to it. So there's first is what we call interphase, which is all this yellow part. This is most of a cell's life. Usually that's how your EOC will like to phrase the phrase interphase as most of its cell life, or where the cell is completing most of its activities. So interphase consists of three different parts. There's G1, there's S, and then there's G2. So, you know, think of the G as growth. Okay, so it's really all that's going on. And then S is important. You'll probably be asked about this specifically. So S stands for synthesis. Synthesis means to make. So specifically, we're making or replicating the DNA in the cell. So again, this is probably like, and it depends on the cell, but this is probably like 95% of a cell's life. Some cells, this might be shorter. Some cells, it might be longer. You know, your skin cells, for example, they replicate like every 3 weeks. So something crazy like that. So that's what dust is. You know, dust is just your skin cells. Everybody's skin cells is dust, if you ever wonder. So now you do. All right, so after interphase, the cell starts going, it starts dividing. So most cells divide through what's called mitosis. There's also meiosis. We'll get into that another time. So mitosis consists of these different phases right here. We'll get into shortly. And then cytokinesis. So, but basically during mitosis, the nucleus is dividing in half. So you'll have two new nuclei, and then cytokinesis is just when the cell actually splits in half. Okay, and then you'll also have some questions on cancer. So cancer relates to the cell cycle. So cancer isn't like caused by bacteria or anything. It's someone's own cells. So someone's own cells are not replicating correctly. So they're replicating at an uncontrolled rate. That's all it is. And usually, so the causes of cancer, there's lots of causes of cancer, but essentially it's all the same. So it's just a genetic defect, a mutation in the DNA specific to the cell cycle. So specific to the rate at which it goes through the cell cycle. And that mutation will cause the cells to go through this quicker than the other cells. And of course, you know, you do it so quickly, it's going to kind of build up on itself, and it's going to form a tumor or some sort of growth. So just keep that in mind. And then that's one of the reasons it's so hard to get rid of, because it's your own cells. So, you know, some of the treatments for this is, you know, I'm sure you heard of chemotherapy. Chemotherapy is a toxin. But what that toxin does is it specifically gets rid of cells that replicate quickly. So that's how it works. But, you know, if you remember, so hair follicles, they replicate quickly too. So that's why a lot of times people going through chemotherapy, they tend to go bald because it kills off the hair follicles. And the stomach lining. So stomach lining is also a type of cell in your body that replicates quickly. So that's why people going through chemotherapy also tend to get, you know, sick very easily, or not, you know, the stomach nauseousness. All right, so let's go ahead and move on to mitosis now. And then All right, so now we covered interphase G1, S, G2, and I briefly mentioned mitosis and cytokinesis. Now we're just going to go into a little bit more detail on mitosis. So first of all, you're also going to learn about meiosis. So meiosis, easy to confuse. One thing to keep in mind, mitosis, think of the TO. I have that underlined on purpose. Think of it as like mitosis because it makes 2 new cells, whereas meiosis is going to make 4 new cells. But anyways, mitosis is cell division for body cells as opposed to sex cells of meiosis. So pretty much any normal body cell, any growth, any repair a cell, any living thing is doing is going to go through mitosis. Okay, so the whole point of meiosis, I'm going to touch on it real quick. I know it's kind of separated, but it just kind of makes sense to talk about them together briefly. Is reproductive cells. So sperm and egg, that's the whole point of meiosis. Okay, replicate sperm and egg cells, and that causes genetic variation. So mitosis, you're replicating your regular old body cells or any living thing. And so you're going to want the DNA to be the same. You don't want different cells with different genetic information. That would not work. That's what meiosis is for. So mitosis creates 2 identical body cells, normal growth and repair. That's just the overall gist of the process. All right, now on your exams, you could be given all the four or 5 different pictures of mitosis, and it could include interphase. I probably should have included a picture of interphase in here. Actually, this top one is interphase. Sometimes it's called early prophase, but this is basically interphase. If you have a nucleus and the DNA is, I call it spaghettified like that, that's just the normal cell. When the cell is getting ready to divide, it's called a nuclear envelope. So basically the nucleus, it just goes away. And then the DNA condenses down into what we call chromosomes. So either chromosomes with X-shaped figures. It's just condensed DNA in a unique shape that makes it easy to move around and separate. Okay, so that's prophase. All right, next stage is called metaphase. So think of the M in metaphase as middle, because those chromosomes, the chromatids, are going to now line up in the middle. Okay, that's pretty simple. There's not much more to it. And then after they're in the middle, they're going to pull apart, or anaphase. So think of anaphase as apart. Its chromosomes get pulled apart. Sometimes it's called the poles of the cell. It might phrase that on your exam, your EOC, your module exam, anything. So keep that in mind. It might call the poles of the cell. It's still the end of the cell. And then kind of going back a little bit, sometimes this is called the equator, the center. So it might not say the middle, it might say the equator. All right, and then after anaphase, you have telophase. So telophase, you have 2 clumps of DNA now, and then the nuclear envelope, the nucleus, starts to reform, except in 2 separate spots. And then those chromosomes uncoil, they get spaghettified again. And then the very last stage is what we call cytokinesis, is just when the entire cell splits in half. Okay, and then just as a side note, so plant cells, very, very slight difference in plant cells during cytokinesis. So that difference, because plant cells have a cell wall, animal cells do not. So the plant cell also has to form a cell plate in the middle before it divides. That cell plate is going to turn into part of the cell wall for each of the new cells. All right. All right, since I'm here, I'm just going to go ahead and run through meiosis too, since this doesn't take too long. And it just makes sense to cover them together. If I have meiosis in this one, maybe I don't, maybe I lied. I'm going to stop recording for a second, see if I can find that.